Hello friends, in this lecture we will study algae based health foods. Algae is defined as heterogeneous assemblage of organisms that range in size from tiny cells to giant seaweeds. Algae mostly are photosynthetic species which include eukaryotes and prokaryotes like cyanobacteria, blue green algae, and they live in aquatic habitats. To meet the nutritional requirements of the rising populations, researchers are looking for alternate sources for food which are easy to cultivate, which are cost effective and which produce large amount of bioactive compounds which are useful for preventing major health diseases etc. And in this regard, algae has a great potential for its utilization in health food development or functional food development. The algae, on the basis of their bioactives present in it, their colors etc., they can be characterized into 4 major groups like blue green algae, cyanobacteria. It is a photosynthetic prokaryote. You can see in the picture here, it is a blue green algae picture. Then the red algae, rhodophyta, eukaryotic algae and is found in the freshwater lakes. Green algae, which is called chlorophyta, it has unicellular and colonial flagellates. The pherophyta or brown algae is branched and filamentous thallus. So, these are the major types of algae and their specific characteristics. The nutritional and medicinal importance of algae lies in its high protein content, good carbohydrate material and their values, vitamin, mineral, pigment, fatty acids etc., which are contained in these different algae. These algae, they enhance immunity, they are hub of the various antioxidants, they have antimicrobial compounds, anti-cancers compound in it, and they reduce cholesterol level. So, they are very beneficial from health point of view. In different countries, algae, different types of algae are commonly used in the food as a food ingredients or even for cooking purposes, for such other purposes like spirulina, blue green microalgae with a long history. It has a long history of use as a food source because of its high protein content and other nutrients. In fact, now in our Indian market also lot of spirulina based product like spirulina powder, spirulina capsule, spirulina tablets etc. are available. Dulse, it is a red species sold particularly in Ireland and Atlantic Canada. Chlorella, it is a popular microalgae, it has similar nutrition provided by spirulina and dulse. It is the purple laver. In Ireland, it is collected and made into a jelly by stewing or boiling. Iris moss, it is the source of carrageenan for the stuffing of instant pudding, sauces and dairy products. Sea lettuce is used in Scotland where it is added into soaps and salads. So, different types of algae in different countries are used for various food purposes. So, in the algae, as I told you that they are the source or hub of bioactives, alright. So, why these algae, there is different algae like Dunaliella, it has a good amount of beta carotene, spirulina, it contains phycocyanins, chlorophyll, beta carotene, Haematococcus, astaxanthin, chlorella, it has lutein, chlorophyll and beta carotene. Porphyra, it contains phycoerythrin, chlorophyll etc. So, various bioactive compounds and are available. So, now the question is why we look at algae as a source of biochemicals or bioactives? The answer is because the algae, it has a short life cycle, it is easy to manipulate and have that the stress conditions in the environment can be easily created to encourage or to get more and more bioactive or biochemical production in the algae. They are simple structure, hence adaptable to different climatic conditions, and they are rich sources of mixed vitamins, minerals and proteins, etc. So, the algae for its use in the food and food supplements, important thing is that is number one aspect is the proper cultivation of the algae. That is for cultivation, like obviously proper practices, standard practices should be followed, all right, for the cultivation of the algae. I will tell you in the next slide what are the different cultivation methods. So, the nutrients required or other facilities required like light, water, carbon dioxide and different organic sources of nutrients and carbon etc. They should be provided, media should be proper media should be provided, and then algae, it should be given required environmental conditions like temperature etc. So, it grows, it cultivates, and then finally, once the algae is grown, then it need to be harvested, that is the taking out the algae from its either cultivation medium, and then this algal biomass, like it is grown in the aquatic media, that is liquid media. So, it is very wet, it contains lot of water in it. So, it needs to be processed, that is the processing of the algal biomass includes dewatering, thickening, filtering and finally drying. So, ultimately that is in this using this the algae is cultivated, taken from its cultivation pond or photoreactor, then water is removed, filtered and it is dried. So, dried biomass now is algal biomass is available for food utilization. So, for its food utilization, two approaches can be made. That is one as such, that is algal biomass directly can be used in food preparation and food formulation. Of course, sometimes this algae they have some very strong flavors. Some people may like, but some people may not like. So, in that case, there is the another approach can be followed, that the bioactives may be extracted from these algal biomass, and these extracted bioactives can be used for the formulation and preparation of the functional foods to provide targeted benefits. So, both approaches, of course, that is the in the case for the case of that is the extraction. Of course, so one has to see that the extraction procedures, extraction technology, methodology etc. should be properly standardized and properly optimized. And for the extraction, one drawback may be there, but that for commercial production, large amount of biomass might be required to get the desired amount of bioactives from this for use in the food etc. But anyway, that is the both approaches can be followed. In the slide, I will take this few example of chlorella as well as spirulina, and I will give you some the food products which are made using these both the extracted bioactives as well as the biomass. So, regarding algal cultivation, as I told you that the microalgae, there are two methods for their cultivation. One is the open pond system and other is the closed photobioreactor system, right. These open different types of open pond which are used, which can be used. There are raceway ponds, shallow big ponds, circular ponds or tanks and closed ponds. But obviously, that is if it is a closed pond or that is the conditions, that the temperature and sunlight etc. all those conditions, it should be optimized and proper environmental conditions should be provided for the algae to grow. So, from those point of view, photobioreactor system appears better the controlled environment of the photobioreactor allows a higher productivity to be achieved, and productivity is the most important indicator for success of the technology behind a bioreactor. So, photobioreactor comes in different designs, different ranges, like tubular type, which is considered to be more appropriate for outdoor cultivation, or flat plate type. The appropriate reactor design is required to obtain the maximum cell mass. The flat plate type bioreactor is favorable due to its low pressure energy consumption, high mass transfer capacity, and high photosynthetic efficiency. So, after the cultivation, then the dewatering of the microalgae, and there are three methods which can be used: flocculation, centrifugation, and filtration. So, flocculation method is used to amass microalgae cells from the broth, and the type of flocculating agents which are used for this purpose include aluminum sulfate, ferric sulfate, ferric chloride, and polyelectrolytes, etc. Centrifugation is generally preferred method for the harvesting of algal cells. It is a most efficient method for biomass recovery as compared to other techniques such as dissolved air flotation or drum filtration. The filtration is the competitive method compared to other harvesting options. Different types of filtrations can be used for getting the biomass, like the commonly used filtration methods include dead end filtration, microfiltration, ultrafiltration, pressure filtration, vacuum filtration, tangent flow filtration, and so on. So, by either using, mostly as I told you, that is centrifugation is considered a better method or easily adaptable method. So, they go for the centrifugation methods, or some in the plants, etc. they also go by flocculation and others. So, depending upon the facility, infrastructure, any of these methods can be adapted, but the important thing that as far as possible, as much as possible, the water should be removed and you get a concentrated algal biomass. And then next step is the drying of the biomass. This after recovery or harvesting through separation of the algae from the support media, the dewatered slurry, the concentrated slurry of the biomass is dried for its stability. That is, obviously the dried biomass is more stable, it is easy to handle, easy to store, all right, and it is easy to use in extraction, all right, or for further processing. So, a suitable drying method and suitable drying process needs to be used, all right. That is because that is during drying process, the bioactives, their functionality, all right, should not be adversely affected. So, the selection of the drying technology accordingly depends on the production scale and also the purpose for which the biomass is being dried. So, different drying methods or dryers includes rotatory dryers, spray dryers, solar dryer, cross flow drying, vacuum self dryers, flashing dryer, or incinerator drying or incinerator dryers, etc. So, any of these depending upon the requirement can be used depending upon the availability of the drying system, but the important thing that the drying process should be optimized and bioactive should remain intact in the dried algal biomass. So, after drying, there is now we have the dried powder. So, it can the powder, that is the algal biomass, dried algal biomass can be sent for the extraction for the bioactives, and then extracted this extracted bioactive as well as spent biomass after extraction, both can be used in the food preparation, or whole biomass can be used as a food preparation, food formulation and food preparation. So, for the extraction, now depending upon the bioactive agent, their solubility in different their polarity, etc. These are taken into consideration while deciding the extraction procedure and the solvent, etc. So, in this slide I have given you like phycocyanins for their extraction, solvent extraction, distilled water or phosphate buffer or calcium chloride solutions, etc. can be taken because they are soluble in water. All right. Then this phycoerythrin, the phosphate buffer, sodium phosphate, sodium chloride solutions, etc. they can be used for chlorophyll because it is soluble in organic solvents. So, chloroform, methanol, acetone, astaxanthin can be solubilized or extracted using ethanol, ethyl acetate, acetone, lutein and zeaxanthin. They can be extracted in acetone and beta carotene, acetone and hexane. So, the important point is that if the solvent to be decided on the basis of their solubility and extraction capacity, and of course, important to say, needless to say that whatever the solvent is being used, that should be food grade, because ultimately the bioactive is to be used in the food preparation. Okay. So, there are different methods for extraction, maybe conventional methods can be used like succinate extraction, freezing and thawing or hydrodistillation methods, or advanced technologies like ultrasound assisted extraction, microwave assisted extraction, enzyme assisted extraction, pressurized liquid extraction or supercritical fluid extractions can be used. Obviously, that is advanced methods, they provide the as far as the functionality of the bioactives, etc. are more, sometimes they are rapid. So, in this here I have given you a brief comparison of the method like ultrasound assisted method is a easy, rapid and economical method. It gives more recovery of the bioactives. Freezing and thawing method, it causes less denaturation of the protein or enzymes and other, but it is a time consuming method. Microwave assisted, it is of course a rapid method, but some heat generation takes place and this heat may have some influence and effect on functionality of the bioactives, etc. Supercritical carbon dioxide method is considered a very good method, green technology. It is a rapid, inexpensive method, but here also that it has some issues with the polar molecules which are present in the. So, one has to cautiously choose the appropriate extraction methodology, standardize these process parameters for maximum recovery of the bioactives, and once these bioactives are extracted, recovered using appropriate method, then this need to be characterized. So, some case studies I tell you that on the basis of our own work, that is the extraction of lutein and zeaxanthin from chlorella. So, this for more recovery, the first the cell is need to be disrupted. So, cell disruption with alumina, then it is given alkali treatment for membrane, that is the cell membrane breakup, etc. That is with potassium hydroxide, and then it is followed by solvent extraction with using food grade hexane. So, we in our studied different solvents, all right, and we found that ethanol gave highest extraction yield, that is 90% extraction yield as compared to other solvent like acetone, hexane, water and various concentration of aqueous ethanol. So, 90% ethanol gives better. Okay. Then extraction of alginate from Sargassum muticum, right. So, in this case Sargassum collected from the sea, they dried using suitable technologies, and then next is the soaking of the dried algae in 2% formaldehyde solution for 24 hours at room temperature, after which that they are washed in distilled water and water containing 0.2 molar HCl, then it is followed by extraction with 2% sodium bicarbonate for 3 hours at 100 degrees Celsius, and finally collection of the soluble fraction by centrifugation, its precipitation in ethanol, and then drying at 65 degrees Celsius until it reaches the constant weight. So, in this way alginates are extracted from the sargassum, and in our experiment we could get 13.5% alginate yield weight by weight. So, as I told you, that is after their extraction, the important thing is that these biomolecules or bioactives, they should be characterized, that whether what is their purity level, whether the bioactives which was needed, that has been extracted or not. So, different characterization techniques can be used, like HPLC, UV vis detector, HPLC dead, or HPLC equipped with a fluorescence detector, HPLC, ESI, MAS, GCMAS, or GC fitted with a flame ionization detector, thin layer chromatography. So, details are given, that is depending upon the compounds, that is the for tocopherol, it is better to have a characterization by HPLC equipped with a fluorescent detector. So, carotenoids, HPLC dead can be, beta carotene can be by HPLC or UV vis detector. So, they should be properly characterized, and then this characterized extracted and characterized biomass should be used in the food formulations. So, now I will give you some of the food products which are developed using these extracted bioactives as well as different biomass in our laboratory. So, one is the ready to serve spirulina beverage. As I told you, the spirulina is a very good algae, and it has been given accorded GRAS status for use as ingredient in food at a small level, maybe 0.5 to 3 gram per serving, and this whole spirulina powder, tablet, etc. are available in the market. So, in our laboratory we formulated ready to serve a spirulina beverage, all right, because the regular consumption of spirulina fortified food improves immune system by increasing phagocytic activity of macrophages, stimulating the production of antibiotics and cytokines. So, four variants of the ready to serve beverages were used, all right. The common ingredients used in the preparation of beverage included sugar as a sweetener, salt, citric acid, some preservatives, and some flavor. Their composition is provided in this, and the variations, that 4 variations in weight by having different proportions of pectin as well as aloe vera in this. So, the pectin and aloe vera concentrations were varied with 1% and 0%, 1, 0 and 10, 0, 0 and 1 and 10. So, different combination and permutation were used, and all these were using standard beverage making, this were made. In this process flow chart, I have just tried to give you an overview, that is schematic of the process, how the beverage was prepared, like Spirulina plantasis variant loner was used. So, this here up to here, that is in our laboratory, we had some bioreactor in which this was grown, all right. Media, CFTRI media was given containing sodium nitrate, magnesium sulphate, calcium chloride, pH was 10. So, it was grown for 16 days, all right, at temperature 33 degrees Celsius, that is 67.5 per square meter per second. Light intensity was maintained, and the biomass was cultivated, it was harvested, and then this biomass was sent to the extracted, that is the bioactives were extracted with a non-polar, that is hexane, ethanol mixtures and other components, and this was followed by vacuum concentration, pre-freezing and freeze drying, and then spirulina extract powder was obtained. So, this spirulina extract powder, all right, which has about 0.47% moisture, containing 0.47% moisture, all right, and this was used in the preparation of or formulation of RTS beverage, all right. So, per 200 ml of the beverage, 0.2 gram of the extract powder, 1% pectin, 0.1% salt, 20% sugar, 0.25% citric acid, 0.5% sodium benzoate, etc. were added. And then we studied, in fact, pectin is 1% and pectin and aloe vera in different proportions in all the 4 beverages were prepared, and they were stored for 2 months at 5 degrees Celsius or at 30 degrees Celsius and 45 degrees Celsius for 30 days, and we studied different effects. So, this is the content characteristics of the ready to serve spirulina beverage, that is its properties which is prepared. This slide shows that is the prepared RTS beverage in our laboratory. Alright. So, it has TS content around 18.6%, coliform count nil, beta carotene 0.22 milligram per 100 ml, chlorophyll 1.100 milligram per ml, and its energy value per serving was around 160 kilocalorie. So, these are the properties of the prepared RTS beverage. We got conducted sensory evaluation and changes during storage, etc., and we found it was good. In fact, this has anti-cancerous and other anti-mutagenic properties, etc., were analyzed, and we found it to has a very good health promoting effect. The other product is the spirulina fortified Srikhand. That is, Srikhand is an indigenous fermented and sweetened milk product. It is regularly consumed in Indian states of Gujarat, Maharashtra, and certain parts of even Karnataka, Madhya Pradesh, and Rajasthan. It has a typical semi-solid consistency with a characteristic smoothness, firmness, and pliability. So, it is consumed directly after the meal, or even sometimes some people use it as a, that is, consume it with chapatis or fried chapatis, puris, bread, etc. So, here it is the process flow chart for the preparation of the spirulina incorporated Srikhand. That is, the spirulina is Srikhand is prepared that as per usual standard procedures, and then this Srikhand once is obtained. In this we conducted a study by incorporating spirulina powder, right, dried spirulina powder, whole biomass, that is maybe point up to at different level 0, 0.5, 1 and 1.5% weight by weight, right, and these were properly blended, homogeneous blend was made, and they were stored for 5 degree plus minus 1 degree Celsius for different period of time to study their effect. Right. So, this you can see in the picture, that is, this is the photographs of samples prepared in our laboratory. That is, here is the control Srikhand, no addition of spirulina, but this B, C and D contains spirulina by 0.5%, 1% and 1.5% weight by weight. So, in our storage studies or effect of this addition and sensory and other characteristics was done, and it was finally found that Srikhand with 1% added spirulina was liked by the consumers, because beyond that it has some issue, the color and the flavor were some issues the consumers did not, because increasing the spirulina content had some effect on the color, maybe darkening of the Srikhand, and that where some consumer did not like, that is panelist did not approve. So, 1% addition of spirulina you could found sufficient, and it did not have any adverse effect on sensory and other characteristics. It contained 1.34 milligram per 100 gram beta carotene, and phycocyanin and chlorophyll A and chlorophyll content also were very good, right. So, addition of spirulina into Srikhand also diluted the acidity, and as a result the pH of this was increased. So, it has some, because 1%, up to 1% there was not significant change, but beyond 1% it did have some effect. Then chlorella fortified pasta. Again, pasta was prepared using durum semolina flour, 2% chlorella biomass and water. The twin screw extrusion method was used. Standard procedure of pasta making was followed. The temperature in the extruder was 50 degrees Celsius. Feeder speed was kept at 25 rpm, and the pasta was obtained. It was finally dried. You can see here in the picture is the fortified chlorella pasta, and then the cooking characteristics and other characteristics of this pasta were studied, and it was, you can see here in this table, the experimental result, and said that the optimum cooking time, cooking loss, swelling index and water absorption was little bit higher or almost similar in different samples of chlorella fortified pasta as compared to control sample. Chlorella biomass fortified pasta contain xanthophyll 0.65 milligram per 100 gram in our experiment, as well as beta carotene 0.163 milligram per 100 gram of pasta within our experimental limits. So, means that this is indicative that people can make, that these products can be commercialized and made. This chlorella fortified pasta, and so our study has demonstrated. Similarly, we have used, that is the beta carotene extracted from chlorella biomass. It has been used in the for the fortification of apple jam. So, apple jam is prepared using a standard process, a standard technology, and towards the end, before filling into, that is once the concentrated jams after it has been taken out from that heating source, alright, before setting, just at that point it is added and mixed properly, that is addition of beta carotene extract when temperature reaches to around 55 degrees Celsius, and then finally it is allowed for cooling, setting and then storing. We analyzed different that is physicochemical sensory characteristics of apple jam, all right, like that adhesiveness, hardness, deformation at hardness, color like LAB values, overall sensory color and appearance, sensory scores, flavor and overall acceptability. So, both instrumental methods were used as well as sensory methods were used, and it was found that the data results almost indicated. In fact, the hardness almost it increased little bit by addition of that chlorella, beta carotene, that is deformation at hardness values, they were almost same. LAB values, there were not very significant change, simply A value there was little A star, okay. B star and L star almost similar, that is color and appearance not much difference, okay. Flavor also, and even overall acceptability, that is it was liked, it the same 7.8 as 8 and 7.6 with the overall acceptability scores. So, the vitamin A content in the control was observed to be negligible, whereas the sample with extract addition had 5 milligram per 100 gram jam, that is the contain 0.13 milligram vitamin A per 100 gram of jam. So, vitamin A content was, or pro-vitamin A content of the jam was significantly increased. Okay. So, these a few products which we have told you here, included here in this, these are the indicative, that is yes, this gives a quite good reasonable data, that is these processes can be developed, they can be commercialized, and it will increase the algal based food products can be prepared. And so, in the finally, I would like to say that this algal based usefulness of the algae in the food industry, like the natural products derived from algae, which may include proteins, pigments, sulfated polysaccharides, alginates, carotenoids, polyphenols, flavonoids, omega 3 and omega 6 fatty acids, minerals, and other essential nutrients. So algae have of all these. So microalgae derived additives like luteins, zeaxanthin, beta carotene, they have huge application in pharmaceutical industries and food industries as a natural colorant and natural antioxidants. Alginates produced from macroalgae are widely used in bakery, ice cream and pharmaceutical industries. During addition of algae or algal extract, the level of incorporation should be such a way that the product's unique sensory characteristics should not be compromised. The technology of fortification or the fortified product should be economical. That is, so some marine algae contain some toxic substances which create adverse effect to human beings. So the selection of algae for food purpose should be scientifically examined. So I can say that in future the algal biomass can be represented as a potential source for nutraceuticals or antimicrobials and antioxidants. Of course, importantly, if the industry commercializes the extraction technologies also, but a proper there is a systematic approach maybe has to be taken, that is the mass cultivation of the algal biomass, because the availability of the algal biomass for the extraction purposes is a big issue, major challenge. Large amount of dried biomass need to be made available to the extraction industries. So having a proper well planned systematic approach of algal cultivation, its dewatering and drying, then extraction, and finally use of these extracted bioactives or the biomass obtained after extraction or even whole biomass as such, they can be used to prepare various functional food products, health food products or food containing various ingredients. I have already shown some of these successes made in our laboratory and some products which we have prepared. Those products, they have very good health value and nutritional value. So such products, they are indicative, they indicate that several such products can be developed, can be formulated, or even these products industry can commercialize and make. So if they are done, obviously these provide a very good alternative source of the protein, and this algae can be easily cultivated, and it can provide a major boost in the protein requirement of the body, and as a protein source it can be used. With this, I thank you very much.